Hello everyone, and welcome to this a little bit different flipped activities review. Typically we would do this in the classroom setting, but in the interest of time we didn't really get a chance to do that. So what we're going to do is that we'll have these animations that you will watch, and I will tell you to pause the video at certain times to watch these animations. And where you could find these animations are on Blackboard. So I'm going to take you to exactly what folder these animations are found in. So what we'll do is we'll go ahead and, just as you would normally, open up Blackboard. As soon as it opens up. Well, since it's taking forever to open up, what you would go to, which is go to Course Content, and then at the bottom of Course Content is a folder that's called, still doing that, it's a folder called Microbiology Animations, and then in that folder at the very top are what are called Chapter 7 animations. So that's where you would find this. All right, welcome back to our Chapter 7 flipped activities. So what we are going to do is there are animations here, and I'm going to have you watch these animations, and then we're going to go over the answers to this. So first things first, let me show you where these animations are. So I'm going to share my computer screen with you. All right, so if you go into the course content folder, you go into that course content folder, what you will find is at the very bottom you have microbiology animations, and then at the very top you have the Chapter 7 animations here. So if you haven't already done so, go ahead and pause the video now, and then upload. Go ahead and go into Blackboard and start loading this page so that you can have access to the animations and watch them. So let's kind of leave this page open, and then we'll go through all 7 of these animations and the answers to the questions here. So go ahead, and I'm going to pause the video now. Go ahead and mute pause the video now and load up on Blackboard. All right, welcome back. So that first animation there is talking about DNA replication. So big things that I want you to take from that is A always pairs with T, C always pairs with G, and T to A and C to G, vice versa. So that's always going to take place. In replication, we call the semi-conservative model of replication because we're going to conserve one half of the DNA strand and then make the template off of it. So we learned in Chapter 6 that one cell becomes two cells on there. So as a result, when that DNA makes a copy of itself, it's going to result in two daughter cells. Now at that point, those daughter cells are genetically identical to one another as well as the paternal strand because these copies its genetic information. However, we know that bacterial cells are not going to always be genetically identical to one another because there are different things that can happen in horizontal gene transfer. So on page 1, part A, what are the products of the semi-conservative replications in a double-stranded DNA molecule? C. Two double-stranded DNA molecules, each consisting of one paternal strand and one daughter strand. Part B, why is DNA replication essential for a cell? D. An organism must copy its genetic material information onto its offspring. That's the whole reason for this song and dance. We want to have progeny in the next generation. Part C, what is the function of paternal DNA in replication? That paternal DNA serves as a template. It's what we call the semi-conservative model of replication. We conserve one strand and make a new strand off of that template. So part C, letter A, it serves as a template for DNA replication is your correct answer. Which of the following is a DNA strand complementary to C, G, A, A, T, C, A, G, C? Well, remember, C goes to G, A goes to T, T goes to A, G goes to C. So A to T, G to C, always in DNA. So if you do that, then what we have for our complementary strand would be letter B. G, C, T, T, A, G, T, C, G. All right, so let's go on to our next animation. Go ahead and pause your video and watch the second animation, which is going to go by transcription. And welcome back. Wasn't that an interesting video? So transcription and translation are an integrated process by which we're going to build proteins. Very important to understand, and this is not just true in microbiology, but in all biology, protein synthesis is the method by which the wishes of the DNA are carried out. The DNA just is like a crime boss. It has desires and things that it wants done, but it never gets its hands dirty, right? So if this crime boss puts a hit out on somebody, they're not going to actually do it, right? They have their henchmen down the line to do it. So same thing with the DNA. It doesn't really have to do it itself. It just gives instructions for doing it. And those instructions for doing it are these two integrated processes called transcription, which we watched the video over, and translation. So during transcription, we're going to get the message from the DNA. We're going to transcribe it into a series of nucleotides. In this case, G still goes with C, and C still goes with G when we go from DNA to RNA. But there's a difference here. In RNA, there is no T. So that T is replaced with U for uracil. So then A will pair with U on the messenger RNA strand, but T on the DNA strand can still be A on the messenger RNA strand because it's still an adenine on A, the messenger RNA. So we make this messenger RNA strand that is then going to be used as the messenger, carry the message, hey, how the DNA was supposed to be built, to do the next process, which is translation, and build that protein. So all of the things that are going to be needed by the DNA or by the cell are going to be given out as commands by the DNA. So in part A at the bottom of page 1, when it says transcription produces which of the following, what it's going to make is, and this is just directly from the animation, is F. Messenger RNA, tRNA, rRNA, they're all going to be produced by transcription because even though they are needed in the process of transcription and translation, they are still instincted by the cell that has to be made under the wishes of the DNA. Part B, according to the animation, which of the following makes messenger RNA or mRNA from the information stored on the DNA? This is another one of the polymerases. We saw DNA polymerase in the first animation when we're trying to make new DNA strands. Now we're looking at RNA polymerase as it makes the messenger RNA strand. So part B, correct answer is letter B. Part C, ribosomes contain which of the following? Well, this is kind of a dead giveaway. It's right there in the DNA. R, and the rRNA stands for ribosomal RNA. So letter A is your correct answer for part C. Part B, which of the following is involved in bringing amino acids to the ribosome? So that transfer RNA or tRNA, what it is transferring is transferring on one end of it the corresponding amino acid to the codon of the messenger RNA, and on the other end of that tRNA, you're transferring the anticodon. So some terms that you want to keep straight here. Amino acids build polypeptides to build the protein. Whenever you hear amino acids, I want your brain to start thinking protein. Another term to be familiar with is anticodon. The anticodon is going to be on the tRNA. It is the opposite or anti of the codon. And then that brings us down to the last term I want you to really understand. Codon. Codons are the sequence of genes in 3 letters, so it's like AUG, CCC, UUU. Those letters correspond to a specific amino acid. The codon is always found only on the messenger RNA, the anticodons on the tRNA, and the amino acid is also carried by the tRNA. So when it says which of the following is involved in the amino acids to the ribosomes in part B, that correct answer is A, transfer or tRNA. Part B, which of the following can be translated into a protein? Only what's carrying that codon can be translated into a protein. So for part B, the correct answer is letter C, messenger RNA. Part A, which of the following statements concerning transcription is true? A, all types of RNA in the cell are synthesized by transcription, which is the source of DNA as a template for copying. That sounds exactly like making a messenger RNA for that process of transcription. B, promoter sequence to the end of a gene. Well, we can stop right there because we know promoter sequence is in the beginning. C, during transcription, entire chromosomes are copied because it starts within the gene that knows. We also know that is incorrect because the DNA can point exactly to where the gene needs to start and stop. D, transcription is where identical copies of DNA are made. We can stop right there. We also know that it's not true. So our correct answer is letter A on part A. So on to our third animation, translation. So remember we said transcription and translation were the 2 integrated processes by which the wishes of the DNA are carried out to make the proteins that are responsible for doing whatever it is the DNA wants it to do. So go ahead and pause this video, and watch the animation. Alrighty, welcome back. So, in translation, what we're getting out of this is a polypeptide of protein. Remember I told you when you see amino acids, it's polypeptides and protein. Protein is just a street name for a polypeptide. So, what comes out of the translation is B. In page 3, part A, a polypeptide. What comes out of translation? Part B, how is bacterial translation different from eukaryotic translation? Well, in eukaryotic cells, they have a nucleus, and the whole process of transcription happens in the nucleus. So after the messenger RNA is made, there has to be what we call post-transcriptional modifications. There's some changes. And those changes included splicing out the introns and that information stayed in the nucleus, and then the exons were spliced together and they exited out of the nucleus where translation took place in the cytoplasm of eukaryotic cells. In prokaryotic cells, they don't have a nucleus. So they have this advantage of ability of being able to have transcription and translation happen virtually simultaneously. So letter A in part C says move in a 3 to 5 prime direction. I'm sorry, I skipped one. Part B is how is bacterial translation different from eukaryotic translation? A, messenger RNA is read 3 prime to 5 prime. No, it still happens 5 to 3 in both the bacteria and eukaryotic cells. Letter B, bacteria can begin translation before transcription is terminated. That is absolutely true. They can happen simultaneously. So letter B is your correct answer for part B. Part C, according to the animation, ribosomes move along the messenger RNA in what direction? Always the same way, 5 prime to 3 prime. Same thing with DNA polymerase. You use 5 prime to 3 prime in that direction. All right, so the next video that we're going to watch here, the fourth video, is over operons. There are 2 types of operons that we're going to talk about, or this video is going to show. You have lax operons, and you have inducible operons, and you have repressible operons. Lax operons are the most notable. Inducible operons are the most well understood, and then trip operons are kind of like our most well understood repressible operons. So we'll take this opportunity to pause this video now and go ahead and watch the fourth animation over operons. And we are back. So, part A, which of the following genetic elements is transcribed into a single messenger RNA strand? What we are trying to transcribe are those specific genes that are going to be required to make this thing happen. So with the lac operon, it only gets turned on, that's an inducible operon, it gets turned on in the presence of allolactose, which means that lactose is present. Usually lactose isn't present, so there's no need to make genes to break down lactose. So when lactose is present, then it turns on that lac operon to make those structural genes. So the correct answer for part A on page 3, which of the following genetic elements is transcribed into a single messenger RNA? That's C, the structural gene. Another thing I want you to really understand is that lac operons or inducible operons are normally turned off unless they're turned on by something, which is usually the presence of an entity that needs to be broken down or needs the genes to be made, the enzymes to be made to break it down. So, and the easy way to remember that is that if you've ever heard of a pregnancy or a labor being induced, that means that the labor is turned off and we want to get it started. So we want to induce it in that way. So inducible operons are normally in the off position unless they are activated. With repressible operons, they're normally on and they're doing their thing until you have enough of your byproduct that's made that you need to turn it off. So repressible operons are always kind of doing their thing and then they have to be deactivated. So repressible operons are normally on and they have to be shut off. Going back to our labor example, if we want to repress someone's labor, it's already going and we want to turn it off. So with that in mind, part B on bottom page 3, which operons are always transcribed unless deactivated? They're always turned on unless we turn them off. That would be D, those repressible operons. Top of page 4, part C, which operons are never transcribed unless activated? Those are those inducible operons. We have to turn them on. Part D, according to the animation, where on the DNA strand is the repressor bind? And that repressor is going to bind to the operator sequence. So if the repressor binds to the operator sequence, that's what happens. So it binds to the operator. So part D, the correct answer is letter C. All right, going down to our 5th animation here, we're going to talk about conjugation. So the next few animations that we're going to watch are going to be in reference to what we call horizontal gene transfer. So remember before we said when we had beta replication take place in binary fission, that those progeny or those daughter cells were virtually identical to one another because the DNA just made a copy of itself. Where in horizontal gene transfer, this is the way that bacteria can pick up genetic information within that same generation, so they can pick up genetic information from their neighbors. So there's 3 different types of horizontal gene transfer we're going to talk about. We have conjugation, which requires direct cell-to-cell contact, transformation, where bacteria can pick up just genetic fragments in their environment, and then transduction, where a virus will carry genetic information. So let's go ahead and watch this first video on horizontal gene transfer, conjugation. So this is video number 5. All right, and we are back. So part A, bacterial conjugation is often referred to as bacterial sex. Why is this term inaccurate? Well, unlike sex with animals and eukaryotic cells, like if you have sex with somebody, you're not going to be genetically changed by the experience. You may be psychologically changed by the experience, but you're not going to be genetically changed as bacteria are. As my grandmother once said, that the only reason that you should be having sex is to make babies or to have offspring. So, with that in mind, part A, letter B, conjugation does not result in the formation of new offspring. So, you're not going to get any new progeny from this bacterial sex. It's just a swapping of genetic information. Part B, what must occur for bacterial conjugation to take place? Kind of already said this, the video did a really good job explaining this out. Bacteria have to have contact with each other. So we need direct cell-to-cell contact with each other. So the correct answer for part B is letter A. Part C, which statement about conjugation is false? And the statement that is false is part B, conjugation is a process of bacterial reproduction, which kind of goes with, what we said in part A, that we don't get genetic offspring as a result of that. So letter B for part C is your correct answer. Part D at the bottom of page 4, based on the animation, what is transferred during bacterial conjugation? And as you saw in the animation, what we are giving from the donor cell to the recipient cell after this process is the circular gene called a plasmid, so letter D. All right, so let's look at transformation now. So in transformation, what's happening is that the DNA, or the cells, the competent cells, will pick up fragments of DNA that have been left in the environment from other cells, even if those other bacterial cells are dead. Kind of creepy, pretty cool though. So they can incorporate genetic information that's been left over in the environment. So with this type of horizontal gene transfer, does not require direct cell-to-cell contact, which is also the reason why they tell you to take all of your antibiotics even if you are feeling better, or one of the reasons, because those leftover fragments of cells, if you have competent cells available, they can pick up the genetic material of them and can possibly become resistant to your antibiotics. You want to get rid of as many of them as possible. So let's go ahead and pause this video and then watch our 6th video on transformation. And welcome back. So, part A on page 5, competent cells are cells that can take up DNA from the surrounding environment and integrate it into their own chromosome by recombination. That's your C. Part B, so this is where we're going to get into Frederick Griffith's experiment here. Mice that are injected with the R strain of Streptococcus pneumoniae. Well, the R strain stands for rough, and that means they didn't have a capsule around it, so it didn't look nice and smooth when they looked at it under the microscope. It kind of looked bumpy and rough. So rough strains, from the animation, what happened when the mice were injected with the rough strain? Well, letter C, they stayed healthy because their phagocytotic cells could easily grab the hold of them and kill those cells. So no problem with putting Streptococcus pneumoniae into these mice that was not encapsulated, that was rough, they didn't have a capsule around it. So letter C is the correct answer for part B. Part C, what characteristic of the S strain allows it to evade the immune system of the mice? Well, that smooth S strain, remember it was smooth because it had a capsule around it. So because of that capsule, it made it really slippery, so the phagocytotic cells weren't able to grab a hold of it. So the correct answer for part C is D. The cells have a capsule. Part D, what most likely explains the recovery of live S strain cells from a mouse injected with heat-killed S strain mixed with live R strain cells? So from the animation, and if you don't remember this, make sure you go back and watch it. We know that, well, the mice can kill the R strain cells. That's not a problem. And if we kill the S strain cells and put it in the mice, well, that's not a problem either because they're no longer virulent. They're no longer alive. They can't do anything. But what we found is when we mix them together, what happened is that those R strain cells, the ones the mice can normally kill, they can pick up the genetic information from those dead cells that we put in there along with the live cells. So what happened is that those rough cells became smooth cells because they incorporated that capsule gene into their own genetic information and they became encapsulated as well. So part B, correct answer is C. The R strain picks up the S strain DNA, enabling it to produce a capsule. Part E, which finding is most surprising in my Griffith's experiment? Well, the part that's most surprising is letter A. The S strain cells were isolated from the blood of mice infected with heat-killed S strain cells and live R strain cells. So what we put in were live, rough, dead, smooth. And what we got out were live, smooth. It was completely transformed, hence the name, transformation for this process. So letter A is the correct answer for part B. So going on to our 7th and our final animation here, this is over-transduction. So this is our 3rd type of horizontal gene transfer. Does not require direct cell-to-cell contact. Is not going to be the bacteria picking up, genes from the environment. It's going to involve a virus taking the genetic information from one cell to another cell. And that virus does it inadvertently. Remember we talked about viruses that they don't have the machinery to replicate on their own, so they have to hijack the machinery of a host cell. And sometimes when they hijack those bacteriophages, which are just fancy words for bacterial virus, sometimes with phages, when they hijack the machinery of the host cell of the bacteria, they inadvertently pick up bacterial genetic information. And then when they go to another cell to hijack its machinery to undergo viral replication, it's going to insert new bacterial DNA into the new recipient cell, and that recipient cell can incorporate that DNA into the cell, and that process is transduction. So we have 2 different types. We'll talk about generalized and specialized, and let's go ahead and watch the final video. All right, and we are back. So on page 6, part A, what is unique about transduction compared to normal phase infection? Letter A. Transduction transfers DNA from the chromosome of one cell to another. Part B, how is generalized transduction different from specialized transduction? This is just something you have to pick up from the video to memorize it. Generalized transduction is initiated during the life cycle of a virulent bacterial phase. Specialized transduction just happens in the lysogenic cycle of a temperate bacterial phase. So letter D is the correct answer for that one for part B. Part C, a transducing phase, A, consists of fragments of host DNA instead of viral genome on it. So instead of, hijacking the host cell's machinery and making new little viruses on there, and having viral genetic material in it, it's going to have bacterial genetic information. So letter A for part C. Part D, when a transducing phase interacts with a new host cell, then C, the DNA from the previous host can recombine with the new host cell, and then that cell can be genetically altered or changed. Part A, which of the following is a method of vertical gene transfer? So conjugation, transformation, and transduction are all horizontal gene transfers. So vertical gene transmission is cell division, binary division, going from one cell to two. All right, on page 7 here, part A, Frederick Griffin did experiments in genetic transformation. Which of the following statements about his experiments is accurate? B, Griffin found that DNA released from dead virulent bacteria enters a live avirulent bacteria and changes genetically, causing them to become virulent. So at the bottom of page 7, we had that whole discussion about conjugation and USDA affecting this meat. So when we go to page 8, I'm not going to read all that. You guys can use that if you like. It says, each description below illustrates one way that genetic information is shared between cells. Categorize each item as horizontal or vertical gene transfer event, and then drag each item to the correct bin. So it should look something like this. Horizontal gene transfer, getting a single new gene sequence from outside of the cell, transformation. Infection by a prophase containing a partial bacterial operon, transduction. Uptake and insertion of transposable elements carrying foreign genes. So those are all considered horizontal gene transfer. Vertical gene transfer, mitosis, binary fission, two haploid cells using the polydiploid cells, those are all considered vertical gene transfer. All right, so at the top of, I guess this would be page 9, it says, consider the following statement. Bacteria can exchange genetic materials through conjugation without the need for any protein structures or control mechanisms. Is this statement true or false? It is false. Remember, in order for bacterial conjugation to take place, we have to have the genetic material travel through this sex toy. And then in bacterial conjugation, we have to have direct cell-to-cell contact. So it's false. So that answer is false. Going down to part C, the expression of newly acquired genes. Why would this interfere? Well, so the question is, let's just read the whole part. DNA sequencing of E. coli colonies is isolated from molecules that have biofilm growth in both bacteria, shows that conjugation has occurred. However, the transformed strain of E. coli do not express any proteins. Last analysis showed that the gene from the L-monocytogen protein is incomplete in E. coli. Sequence comparisons show that E. coli has a truncated version of the gene that begins at the nucleotide number 54 and continues to nucleotide 1257, meaning that there is no start codon present. The fact that there is no start codon present, why would this interfere with transcription and translation? Well, the last two bullets there. There's no promoter region for the transcription to begin at, there's no start codon, and there's no start codon for translation. So that's why we can interfere with it. So then our last question, well, not the last question, but the next to last question on here. It says, order the following steps from transcription to the initiation of translation, and here you have it. So go ahead and pause this video right here to look at this. And then finally, our last question in the back on page 10, translational problems. If the inserted sequence had been complete except for missing the last 54 nucleotides and thus the stop signal, would a working protein be possible? And the answer to this is yes and no are probable. If the DNA sequencing after the inserted gene needs to be done to see whether or not any stop signals will be correct for the reading frame or possible amino acids. So really what that's saying in letter B is that maybe not. We have to exactly see if those essential portions of the amino acids are already on there, then it's okay if it stops a little bit short. If the other ones aren't as important, aren't as essential, then we could probably have a usual protein. If, however, it's kind of the essential amino acids are kind of sprinkled all throughout and you're missing some of those essential amino acids, then no, you will not get a protein that works properly. All right, so that is the end of all of this. The correct answer for part E is letter B. And I will see you all in class very soon. Have a great morning, afternoon, or night. Bye-bye.